Dr. Bhardi Pandey, he's a senior scientist at Integrated Laboratory Solutions, North Carolina. And he has been at various association as a senior research scientist, to name a few, Cambridge, Edista Pharma Solution, Synexis, and also as a research scientist in Synexis. And regarding his education, he has done postdoctoral research at North Carolina State University, and also another postdoctoral research at Tulane University under the guidance of at another alumni of our college, Tayi Manwan sir. And he has done his PhD at University of Hyderabad under the guidance of a stalwart of a department, Professor Periyasamy. So he has numerous number of publications and also so many patents to his credits. Just one which I saw in his resume is he has recently patented his pyrocyclic compound as Janus kinase inhibitor, which is used as a therapeutic application for cancer and inflammatory disease treatment. Proud to have you here, sir, with us. Now I would like to open the forum for you to have your discussion, sir. Thank you very much for accepting my invitation and joining us, sir. The floor is open to you. Thank you, sir. Welcome, sir.
Thank you very much. Can you see my screen?
Yeah. Very much visible, sir. Very much. You can proceed, sir.
Okay. Good morning or good evening, everyone. Thanks for your nice introduction. And it's my pleasure to be back with you all, with American College alumni and everyone in the chemistry world. I made this content of the topic mainly for the master students. For the experts, those who are there, please bear with me. It may be too empirical for you guys. So my experience in the drug discovery, and also continue my current focus on the work related to the COVID, I thought I would share what I have been closely watching in the field of the drug discovery, particularly the antiviral drug discovery towards the COVID-19, which has brought a pandemic globally. So most of the global pandemic is caused by infectious diseases apart from the man-made one. So those pathogens are virus, bacteria, fungi, and parasites. Particularly virus is still threatening the humankind, and we do not have a biggest arsenal to fight back with them. So looking at close structure content of this bacteria and virus. So looking at the bacteria is a well-defined cell which has its own machinery to reproduce itself and survive itself. Plasma membrane, cell wall, capsule, or cytoplasm with other ribosomes, which is an important machinery for it to reproduce things. And on the other hand, virus, which is nothing but a DNA or RNA genome kept inside the protein capsule, which needs a host cell to reproduce itself. But it has definitive structures, though, like fusion proteins, attachment proteins, and the viral RNA and lipid bilayer. So the viruses, based on their structures and their constituents and their attacking the target host, they are classified into many families. As you can see, there are different kind of viruses. So the viruses that cause the COVID-19 is a family of coronaviridae virus. It's called coronavirus. So as you can see, different viruses cause different disease, like HIV attacks the immune system, and hepatitis B attacks the liver. The coronavirus attacks the lungs and causes the severe acute respiratory syndrome. So, replication process. See, some of them I have shown here is the protease or the host signal proteases. So all these proteases will help the virus to become mature and release it in the host cell. So pretty much the virus is the smartest thing. I always say that. It enters into your body, someone entering into your house and use all the infrastructures and survives, and use all the human machineries to replicate itself and goes out of the cell. So looking at this, the genome and the proteins, so the, the drug discovery scientists look at what are the like a target that we can kind of interrupt to stop the replication of RNA-dependent RNA polymerase, which is going to help the virus to make the replication of the genome. So I'm going to walk through what are the drugs that have been like advancing in all these four proteins. So the first one is the spike protein. The spike protein is the one that attaches to the receptor, is a angiotensin converting enzyme 2 receptor. So that helps the viral entry into the cell. The another protein, which is the transmembrane protein, actually activates the ACU2 protein. So the scientist trying to kind of attack this transmembrane protein to stop the beginning entry of the virus. See, one of the compound which is already in the market as an anticoagulant drug, nafomostate, is showing pretty good activity against this enzyme. And I'm sure that this is already in the clinical trial to stop the viral entry of the coronavirus. There are many of such like inhibitors are in the trials, and this is the one that is most advanced in the most advancing stage. The second one is a cysteine protease. So as I mentioned before, it cuts the long protein into 11 places to make the other essential viral particles. So interestingly, this protein has a close structural features to the HIV protease inhibitors. So the scientists are kind of screening all other known protease inhibitors to inhibit this protein. See, the two drugs, lopinavir and ritonavir, are advancing and showing a promising inhibitory activity towards this cysteine protease. And also, they have done some screening. Those screening has brought up some beta ketoamide inhibitors. So the advancement in these things, the people were quickly able to crystallize these proteins and do some computational modeling through the virtual screenings. And the things that you see in the screen is, I'm sure they're like a virtual ligands. They have not advanced to the next level, but there's a promising results that will help the structure-based drug design to go into the next level. 4 basic nucleotides. So these RNA polymerase put these nucleotides together to create the copy of the RNA which comes from the coronavirus. So showing it here again. So pretty much all the nucleotides are converted into the long chain of the RNA backbone using the enzyme polymerase. So this is where the structure design comes in. If we interrupt this polymerase by supplying a chemically modified or intellectually designed a fake nucleotide, that will enter into the backbone of the RNA, that will arrest the further copying of the RNA genome. So that's a kind of an approach that actually led to the discovery of remdesivir. So here you can see I have just put up some of these drugs that has been designed based on this concept of creating intellectually designed synthetic nucleotides that will enter into the genome of RNA or DNA, and that will stop the multiplication of copying of the genome. And the remdesivir is one of those drugs. So remdesivir actually, you can look at it. It's a nucleotide with a sugar and a base and a phosphate group. So basically, the active form of the remdesivir is GS441524. So it gets cleaved in the body system and gives you an active drug, which is actually adenosine nucleotide. So taking a close look at remdesivir, remdesivir is a nucleotide analog with a broad spectrum antiviral activity. It just didn't come overnight. This was actually discovered several years back in order to control Ebola virus. This remdesivir is very active against MERS, SARS, and SARS coronavirus 2 in vitro. And also it was active against the Ebola virus. Unfortunately, remdesivir failed in the Ebola virus human clinical trials and did not advance to the Ebola treatment. So when the COVID-19 pandemic came, so they wanted to test against the SARS coronavirus 2. Luckily, it showed some preliminary, it shows the in vitro activity. So most of you know, the clinical trial data showed 60% cure rate, and they have approved this drug on an emergency basis. And again, this is only approved right now for the patient requiring ventilator. It is not given to anyone who shows the positive symptoms of coronavirus 2 infection. So that's an important point. So remdesivir still is not a first-line treatment for the infection, but it is only for the people who have been in the terminal conditions. So it still requires a broad clinical trials under the healthy individuals, but this an emergency drug has been approved. And one of the things with the coronavirus, the SARS coronavirus 2, it has a proofreading mechanism. That means when it makes the copy, if it finds there is an unusual in the replication process, that unusual is nothing but introduction of a drug into that, it will find and delete it. But luckily, the remdesivir is still like evading the proofreading mechanisms. But eventually, the virus will become more smart. It may bring a resistance to remdesivir, which we may not know right now. So that's one of the drawbacks of the remdesivir. So like remdesivir, there are more drugs. If you look at the structure of the remdesivir, as I showed, it's a phosphate prodrug. And this concept is not new. We have already a similar drug that has been in the market, like sofosbuvir is a similar prodrug. These phosphate prodrugs are pretty much, it's not a new concept. But the idea is to find that magic bullet which has the right base with a pendant group that will make the drug more microstome stable or more stable in the body system. For the people who are organic chemists, I'm just showing here the synthetic process the remdesivir is made. So I'm not going to walk through the whole process. I like to point out, if anyone look at the synthetic process, so medicinal chemists always would like to make that few milligrams to test against the in vitro assays, as we always do that. But it's not easy to translate that same synthetic process into manufacturing. If anyone look at this process, you can see the most dangerous reagent, the N-butyl lithium, has been, and the sodium hydride, again N-butyl lithium, and the temperature is -78. These are not the process one would like to take into the multi-ton quantity manufacturing. So always there is a process development doing this manufacturing of the drugs. So the second generation has made a pretty good improvement where they do avoid the using of the butyl lithium and instead they use the Grignard reagent. But if you look at the previous slide, you will see they use the base as a bromo intermediate where you need a butyl lithium, many inflammatory proteins that destroys the virus. But what happens in this case, somehow this protein, somehow coronavirus created a machinery that that kind of survives the 14 days by cheating the interferon. Actually, the interferon is the one that immediately tries to attack the virus and helps the body to recover. So the coronavirus actually evolved a system to cheat the interferon attack. So interferon is a cytokine, as I told, is helps the body to fight back it. So what happens? Interferon expression is suppressed. Nobody knows how it does. So people are working towards it. But what body feels it, there is something coming in. It starts a different mechanism to create other cytokines, pro-inflammatory or anti-inflammatory, to kill the virus. So a normal immune response creates a macrophage and dendritic cells to release cytokines, and that actually kills that actually makes the lymphocytes to produce more particle to kill the virus. So again, the destroyed virus liberates some more cytokines that releases some lymphocytes. That damaged lymphocytes creates more and more cytokines. Now, this like a high production of the cytokines leads to the attacking the healthy cells, which leads to the acute lung damage. This is the biggest difference between other viruses and the current SARS coronaviruses. So high level of inflammation needs to be suppressed. But the problem is when you suppress the immune system, you make your body more vulnerable to further attacks. So there is a fight, or there is a fine line. A balance needs to be found. How much compromise you can make in your immune system to reduce the inflammatory response that won't kill the healthy cells. So there is a need for anti-inflammatory drugs. So we are fighting like two enemies. One comes as a virus, one comes within our own body, which is a cytokine storm. So the cytokine storm is not just one. As you can see, the TNF alpha, interleukin 1 beta, interleukin 2, interleukin 6 actually is the most one that is actually expressed in most of the viral attacks. It's actually the one needed. But in this case, we have to suppress this one so that it won't kind of lead to the attack of the healthy cells. So there is an approach to kind of suppress the immune activity. So what people resorted to take the anti-inflammatory drugs that are already in the market, or it's being used to suppress the immune system. So one of them is a ruxolitinib. Is actually a kinase inhibitor. It's being given to the COVID-19 patients, and they have seen a progress. The another one, which is already in a clinical trial, tocilizumab, is actually an antibody. So as I mentioned, the interleukin 6 is a cytokine that leads to many pathway signal activations. So blocking that would actually decrease other inflammatory pathways and actually reduces the B cell activation. B cell is a one that actually creates a permanent antibody for the further viral attacks. So basically, stopping this IL-16 will bring down the inflammation down. So that will kind of help us not to get the healthy cells damaged. The another one is the baricitinib. Actually, it is a kinase inhibitor. It's an anti-inflammatory. So people are like looking to kind of rushing to use already available anti-inflammatory drugs. So there is now need for two multi-directional approach, at least a two-directional approach, where you need to have a drug to stop the viral replication or viral load, which is done by remdesivir. And another one, you need to have an anti-inflammatory drug. So the National Institute of Health already started a clinical trial giving the combination therapy where you give the remdesivir and the parasitinib together. And there is a positive results and the response is being already seen for the combination in this therapy, which is actually a good thing. So the last one is it's a vaccine. So the vaccine, I'm not going to go into the details of the science behind the vaccine, but I would like to share where we are in the vaccine landscape. So vaccine could be created with a live attenuated virus or inactivated virus or some peptide based or virus like bacteria. And instead of giving the actually like most of the vaccines that are created is using the inactivated virus. And a couple of years back, a company called Moderna started a technology from I think MIT professor Robert Longer, who instead of giving the dead virus, he proposed to give the patients just the RNA genome. So the genome will be given into the humans. So the cells will use the genome to create the virus particle that will create the antibody. So instead of giving the virus, so you give the just genome itself. This technology again failed for Ebola virus. So it was advancing, but very recently, probably a couple of weeks back, this Moderna has advanced into the clinical trial. It has shown a positive response. That means the patients have produced antibodies for the genome that was given as an IV. So that is one new technology that is coming up, but it's not successful technology, but it has more promising. The idea is if this success, the manufacturing of the vaccines will be very fast because it is just a genome. Any genome can be made using the automated nucleotide synthesis. But it is advancing, but everyone is waiting on the trial. But so here I have seen there are more than like 115 candidates that went into the studies, and 78 of them are confirmed. That means the study is progressing. Only few of them were progressed into the phase 1. What I'm showing is one from the Moderna, another one from CanSino Biologicals, another Innova Pharmaceuticals, and another couple of them from Genome Immunes. They are definitely into the phase 1 trials. That means it has been given to the healthy individuals. So unlike the small molecules, this vaccine development is going to take longer than we think or we could, because it has to be tested in the healthy patient, and the safety needs to be studied. So it's going to take its own time. So that's where we are in the vaccine landscape. So hopefully nobody's promising very quick, but hopefully we'll have the vaccine coming through. So that's about the small molecule vaccine we are. If I could summarize, the COVID-19 is caused by SARS coronavirus 2, faced with many scientific challenges. As I have mentioned before, and this virus is more potent and efficacious drug still need to be developed. We have not developed a most efficacious drug. It has not been taken into the regular clinical trial yet. The remdesivir has been directly given to the terminal patient. And this virus, as I said, had made a smart machinery. Somehow it evades the innate immunity, and it replicates 50 times more faster than the other viruses. That means even before our body sends, it has already made the copies of itself enough to attack, make more damage. And the new thing, it creates a cytokine storm, which I was talking about. It's really leading to multi-organ failure. and vaccine still under the development, but clinical trials on patients will take longer time. And many so the many companies actually are screening the other drugs. It's called drug repurpose. The advantage of the drug repurpose, these drugs have already been through many trials, has been shown as a safety in the humans. So that's another approach. We are all rushing into that. So that's kind of my overview from as a medicinal chemist and a synthetic chemist point of view. So I'll be happy to answer if you guys have any questions. Thank you very much for listening. And again, I'm really very thankful for American College and Dr. John for inviting me, to giving me this opportunity, and other faculty members who have been working, connecting the people around the globe. And I thank everyone to join and taking time to listen. Thank you very much.
Thank you very much, sir. Current scenario of coronavirus, you have given an insight into the unknown facts about coronavirus. Thank you very much, sir. There are so many questions. It is just floating. The chat box is floating with questions. Maybe I'll pick one or two for your. So maybe the first question from Kartika: What was the disadvantage of hydroxychloroquine, and why was it stopped in use? So that was our question.
So hydroxychloroquine, as I said, I have only presented that has a definitive answers and a scientific support. So hydroxychloroquine has been shown as a prophylactic on, as mean give it to already before you get any infection. It has shown some positive things, but it doesn't have any structural proof or any yeah. So most of the drugs that we target is one of these proteins. These proteins are made of the amino acids. So what happens when these virus find that it's getting disturbed or interrupt by binding to a particular amino acids, the virus change that amino acids into a different amino acids so that the drug will not bind anymore. For example, if there is a leucine, it will convert into isoleucines or cysteine. So it will change the sequence of amino acids to make the drug not to bind to the protein, which is a typical way any bacteria or virus will develop a resistance against the drug.
Okay. The next question is from Jemima Manoj. She has asked, or she is asking that they're claiming that COVID-19 is only a bacteria, and she's asking, what is your observation in this regard?
I'm sorry, COVID-19 is the name of the disease and caused by the SARS coronavirus 2, and it has nothing to do with the bacteria. Okay, so probably it could, as I said, if we compromise the immune system, the bacteria also will attack us. Yeah.
Okay, so the next question, maybe the last question, because of want of time, I'll just stick to the 4 questions here. It is from Abhilasha Jain. She has asked, one of the Siddha doctor has claimed that he has invented cure for coronavirus. He says a herbal extract medicine is used to treat dengue, multi-organ failure, and similarly acute liver fever. Could this be a right way of treatment, like Siddha treatment can be used, something of that kind? She has been very, does not emphasize the real question, but I think this is a crux of a question. Whether we
I know. Yeah, see, so when we approach a typical drug discovery path, we wanted to have a drug tested for efficacy, safety, and reproducibility. See, most of the Siddha medicines, as we all know, it helps to boost our immune systems. And if we want to show that, we have to take into a regular path of the trial, where we want to show it really reduces the viral load and helps the body to recover. So I understand this. The Kabasura is kind of a mixture of 18 herbals. There could be 20 or 200 ingredients, and we don't know what exactly is going to do us unless it helps to boost our immune systems. Yeah, it can be taken, but we cannot say it is actually doing it unless we test it. And we have to take a typical way of taking it through the in vitro testing. Actually, it's easy to just test. There are like in vitro assays available, coronavirus. All we have to do at least to test the against the in vitro assays. I don't know if those things have been done, but taking those just may help our immune system.
Thank you very much, sir.